All right, welcome back everyone to part two of our general biology lecture talking about energy and metabolism. Um, Earlier, in the previous video, we talked about energy and its different forms and how they're relevant to life. In this video, we'll get into talking more about the chemical reactions themselves. So there are a number of different ways in which we categorize these reactions and refer to them. One is in talking about redox reactions, which is referring to the movement of electrons in these chemical reactions. So redox is just a combination of reduction and oxidation. You have oxidation. Every of one of these chemical reactions has some form of oxidation and reduction going on. So the end chemical that is oxidized in a reaction is one that is losing its electrons. And it is named that because often it's oxygen that is the one that is taking the electron. This is the case in when it comes to aerobic cellular respiration, where glucose is oxidized and oxygen is the one taking the electron. Oxygen is being reduced, it is gaining that electron. So one chemical loses an electron, the other one gains an electron, is reduced. So you may hear the acronym oil rig. Oxidation is loss, reduction is gain. Another example. So oxygen is not always the oxidizing agent, but very often it is. As is the example here in this video, this turned brown.
Let's say you've got an apple, and it looks delicious, and then you bite into it, and you find out it's all gross and brown inside. Or maybe you just want to eat half an apple, so you slice it and leave the other half on the counter. An hour later, it's a brown, mealy mess. All I wanted was an apple, and now all I have is a mouthful of disappointment. What happened? Well, different kinds of cells have different kinds of enzymes, and one enzyme that's found in the flesh of fruit, as well as the flesh of people, is polyphenol oxidase, or PPO. Its job is to interact with phenols, simple organic chemical compounds that can be found in pretty much everything alive. Specifically, PPO oxidizes these phenols wherever it can. That just means that it encourages them to bond with oxygen atoms, and it'll do this as long as it has access to both phenols and oxygen. Now, the oxidation of phenols kicks off a long and complicated chain reaction that ends in a protein you've probably heard of. Melanin. The same pigment that controls your hair and skin color is also produced by fruits and veggies. It's what you're seeing when your apple turns brown. And actually, melanins are a lot more widespread in plant tissue than in our own tissue. We can only really produce them in our hair, eyes, and in the bottom layer of our skin, whereas plants have them practically everywhere. And the melanin that plants make serves a different purpose. So when plant cells are damaged, melanin is produced to surround and slow down any possible infection. That is why, if you've ever cut open a rotten apple, the rotten spot is usually surrounded by browned flesh. It's essentially the apple's immune response to rot. Unfortunately for us, it's also the apple's response to any kind of damage. You drop the apple on the floor, it kills some apple cells and starts cranking out melanin to protect itself. And when you cut an apple into slices and serve them with crackers and cheese, those slices are going to brown really, really fast. Because not only are those cells damaged, but they've been exposed to air, which, as you may have noticed, is full of oxygen, which allows the PPO in those cells to basically throw a big oxidation party. It'll oxidize all the phenols. That apple is going to be so safe. Except it's not going to be safe at all. You're either going to eat it, or you're going to throw it away when it gets all brown and nasty. Because those nice fresh phenols are really tasty, but the melanin? Not so much. But on the bright side, the brown color in cocoa, raisins, coffee, and tea are all caused by this exact same oxidation reaction, and that actually gives those things the taste we like about them. Thanks for watching this quick question, especially to our Subbable subscribers who keep these answers coming. To find out how you can help support our question answering, just go to subbable.com/scishow. And if you have a quick question, let us know on Facebook or Twitter or in the comments below, and don't forget to go to youtube.com/scishow and subscribe.
So again, oxidation is not all bad. Produces some great things, but oxygen is everywhere. It's very high in its ratio in the air, and oxygen as an atom, as an element, is a little greedy when it comes to the electrons. Remember, it's pretty electronegative, so it will readily take the electrons off of other substances. So this is where things get a little bit more complicated in thinking about chemical reactions, we got to think back to entropy and introduce a couple other concepts of enthalpy and free energy. So remember, entropy is that just randomness or disorder in substances that is constantly increasing on the scale of the whole universe. So that's entropy. Then we have enthalpy, which is the total energy that is present within a system, whether that is my biological system as an organism, it could be the system of a cell, or a system of a specific molecular bond. So different scales of systems. Again, could scale that back up to larger biological systems. But when we're thinking about a chemical reaction, we're thinking of the energy present within the system of the chemicals in a reaction. So all transfer of energy involves losing some energy into an unusable form, such as heat. That is what gives you entropy. In a spontaneous reaction, at least, entropy has to be increasing. Again, that's why we need a continuous input of energy into life in order to make the sum of all our reactions, our metabolism, possible. So when we're thinking about this, we have to look at the sort of free energy, which is the energy that takes place, that is available after, or it's the energy for a chemical reaction to take place after we've accounted for entropy. So accounting for the fact that we're going to be losing some entropy, losing some energy to entropy, there has to be enough energy available for a reaction to happen. And if it's a reaction that has a surplus of this energy, then it may happen spontaneously. Otherwise, it will require an energy input. So there's this equation that we deal with where, like, this delta G, that change in free energy, has to be equal to the change in enthalpy, which is often in the form of heat, of temperature, minus temperature times the entropy. So that is our equation, or the change in entropy. So that is our general equation for that. But there are two forms of reactions that we need to concern ourselves with, which are the endergonic and the exergonic reactions. So we have that free energy equation based on how a reaction will change the energy within the system. But there are reactions that have where this equation comes out to be positive, the delta G comes out to be positive, and there are reactions where the delta G comes out to be negative. Now, we're in a chemistry course, we might be calculating this a bit more, but suffice it to say for us that we have our positive delta G reactions and then our negative delta G reactions. We have our endergonic and our exergonic reactions. So the positive delta G reactions are going to need energy in order to occur. So these are anabolic processes, or anabolic processes. These processes that are adding multiple molecules together, bonding things together, that's going to require energy. That energy is going to be stored within those bonds. But for that reaction to occur, it's going to take an additional input of energy. If there's a negative G, a reaction that releases energy, we call that an exergonic reaction. Exerg, that's the outside energy that's releasing energy to the surroundings. So those catabolic processes that break down chemical bonds, break down molecules and their chemical bonds into smaller pieces, catabolic process, those are going to be exergonic reactions where they're going to release energy, thinking of the breaking down of sugars. So bringing that back to the earliest form of these reactions that we thought of, the hydrolysis of carbohydrates. Remember, hydrolysis is when water is helping us to split up a carbohydrate. So if it's splitting up, is that going to be endergonic or is that going to be exergonic? Okay, so splitting up, you're going to be breaking those bonds. The energy that's in those bonds is going to be released. So it's releasing energy. It's going to have a negative delta G. That is going to be an exergonic reaction. Endergonic means it's taking that energy in, it's using up energy. So it should be an exergonic. Bam. So when the thing is, though, there's some energy for these reactions that's required just to get them going. And all chemical reactions can be done in both directions. They won't be spontaneous in both directions. They won't have, a spontaneous interaction, a spontaneous reaction will have a positive delta G, a negative delta G and will release energy. Does not require any external input, but a non-spontaneous interaction with a positive delta G, an endergonic reaction. These all kind of align with one another. I know it's a lot of terms, but those are not going to be spontaneous reactions. They're going to require an additional input of energy. And for those to happen, there will be some amount of energy more than than the overall change in energy, the overall change in the free energy. So what a catalyst does is, remember, a catalyst is a chemical that helps move along reactions. A catalyst is a substance that reduces the activation energy. So it makes it easier for a non-spontaneous reaction to happen. So, like I said, endergonic and exergonic reactions can be either spontaneous or non-spontaneous. What I was saying earlier was more of like a general thing. So the spontaneity of a reaction is not necessarily 100% to do with is not necessarily to do with the sort of energetic favorability of reaction. So like endergonic, exergonic, that's sort of like, is this an energy releasing or an energy using reaction? Spontaneity is a little bit more to do to this activation energy, this additional bit of energy that is needed in order to get things going. So in the case of an endergonic reaction that requires an input of energy, you got a big hill to go up. You got to use energy. And then there's also a little bit of an activation energy to get over for it to roll down to the end of the reaction. In the case of a exergonic reaction, it is going to roll downhill a lot easier, but it still needs to get up over that hill. And what a catalyst will do is interact with the molecules in the reaction without getting consumed in that reaction, but it'll interact with them in a way that will make it more spontaneous and more likely to happen. Remember, none of this also has to do with speed of reactions. This is just sort of the spontaneity, the likelihood, the probability that it'll happen. Oh, is that just a double slide? Okay, so all kind of confusing there, but you have our endergonic and our exergonic reactions, referring to whether they're using or releasing energy, both of which can have some amount of activation energy that is beyond that overall change in energy. There's some other amount of energy that is needed to be overcome in order for them to happen. And then that sort of flow of energy in these reactions involves some transfer of electrons. So as bonds are forming and reforming in different shapes, different arrangements. You're having some electrons that are being lost in the case of a substance that is oxidized, and electrons that are gained in the form of substances that are reduced. So again, the catalysts, though, are important to this, especially when it comes to biological systems. We have our own biological catalysts, our biomolecules that do this. So many of the reactions needed to build that order, sort of fight against the entropy, are high activation energy reactions, but we have enzymes. So enzymes are a specific type of catalyst. They are biological catalysts. They are proteins, typically. And here, we're going to watch a little video on how enzymes are built and how they function. Bring this up here.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur. To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell in every living creature on the planet.
Yeah, enzymes are everything. So we'll keep talking about enzymes because all that diversity of proteins and how they're structured, all their little R groups that they have, they're what enable all of the reactions that are going on, all these complex metabolic pathways. You saw those little webs of the citric acid cycle, and we'll talk about the citric acid cycle, which is key part of cellular respiration. All of those reactions, they require enzymes because otherwise they've got too high of an activation energy, and they wouldn't really happen by chance if there weren't already all these diverse shapes of proteins that can help them along. And also, great little illustration of how an active site is actually doing its thing. Like it's actually gaining and like it's taking pieces off of the molecules it's interacting with, like it is reacting with them, but in the end, a catalyst, including an enzyme, is at the end of the reaction, it returns to its original shape, which means it can keep going. So enzymes, they're made up of proteins. They have a reactive site that facilitates the reaction so that the activation energy is lower in that overall chemical reaction. And then the substrate, which is the piece that it is interacting with, will bind to that reactive site of the enzyme in order to undergo the reaction. So the citrate in that case was the substrate that was interacting with the enzyme, becoming isocitrate. So the enzyme starts where it finishes where it starts. Sometimes you'll see little cartoons of an enzyme just simplified like this, where it's got a perfect little shape cut out, an active site that it will only interact with certain substances. So they're sort of specific in how they interact with things. Some active sites can interact with multiple sorts of things, but there's the part that's doing the interaction, and then sort of like the rest of the structure of an enzyme. So we can think of those interactions like we have an enzyme that is sort of its main function is to interact with a particular substrate, but you can have other chemicals sort of interacting with the enzyme to influence its ability to do that. So we call those inhibitors and inducers. So the inhibitors are the ones that get in the way of the enzyme's normal function. They can do this by completely blocking the active site, like maybe they have a similar structure to the substrate, so they're able to bond with the active site itself. Or they can do this in a non-competitive way by binding not to the active site, but then changing the shape of the enzyme to where the active site isn't really usable, the substrate doesn't fit anymore. But either way, an inhibitor is another substance that is interacting with the enzyme in a way that keeps it from doing its enzyme thing. But then we have inductors. So these are ones that induce the enzyme to actually do something. So we see this in the case of enzymes that act as protein pumps within cell membranes. So an inductor will actually increase the activation or the speed of the enzyme by interacting with the enzyme to change their shape that can then reveal the active site. So this is what makes it so that or alternatively, it can help to guide the substrate to the active site of a molecule. In the case of, it's not the exact case depicted here, but we've talked a little bit in talking about membrane potential, like the sodium-potassium pumps, the protein channels that are needed for that. Is exactly how that works, is when I think it's a calcium sodium pump, where like calcium comes in, it interacts with the enzyme to open up a channel for sodium to move across. But these can be organic or inorganic molecules, so they could just be like ions interacting with the protein. They could be organic, they could be other enzymes, other biomolecules. If they are an organic molecule, we refer to them as a coenzyme. Like they're not the enzyme itself, but they're also sort of like the co-pilot. Like they're necessary for it to operate. But again, they do this by binding with the enzyme to change the shape, reveal the active site. Pardon me, the dog's barking. Reveal an active site, or to redirect the substrate to the active site. So, one of the biggest things involved in this is ATP. We need to go back to our friend ATP and understand it fully, because, again, across all life's organisms, ATP is the main currency for any of these reactions that require energy. ATP is essentially acting as a coenzyme. Oh, wait, that's right. We'll put this on the other tab. Let's learn a little more about ATP.
ATP. We love it. You'll find ATP in so much of our science art, whether it's a GIF of a mitochondrion or cell transport, or a comic about cellular respiration or fermentation, you'll see ATP mentioned. So why the big deal? Why is it all over the place? Many times students will get it in their mind that it is an energy currency of some kind. When I first started studying biology, I noticed that in textbooks, it's often represented like this starburst thing or thunderbolt. And, I guess in my mind, I imagined it was like some big blast of energy that helped the cell do things. And by do things, I mean that we need ATP to do many cellular processes. Examples include active transport, such as when a cell is trying to move something against its concentration gradient, or its role in muscle contraction with the actin and myosin cross bridge. We need another video for that. ATP is critical for many types of cell signaling. You need your cells to be able to communicate. Those are all just some examples. But what is ATP? How do we get it? And how does it work? Those are the basics of what we're going to focus on in this short video. So what is ATP? If you remember the 4 major biomolecules, ATP would fit in with nucleic acids. Yes, like DNA and RNA. ATP is a nucleotide derivative, so it has those 3 parts you'd see in DNA or RNA nucleotides: phosphate, sugar, base, but it actually has 3 phosphates. ATP is short for its full name, adenosine triphosphate. This fancy name is helpful as it tells you that it contains the nitrogenous base known as adenine and 3 phosphates, hence the tri in adenosine triphosphate. Its sugar is ribose. How do you get ATP? All cells need ATP, and so they need processes that can be used to generate it. But the process can differ. It might involve oxygen, such as aerobic cellular respiration. It might not involve oxygen, such as anaerobic respiration or fermentation. During cellular respiration, plants break down the glucose they made from photosynthesis to make ATP. During cellular respiration, animals break down the glucose they consumed to make ATP. And it's not just plants and animals. Bacteria, fungi, protists, and archaea, they all need to make ATP. We have a video on cellular respiration and another one on fermentation that can be helpful to understand the process. But one thing that we do want to mention about making ATP is it's important to understand it is part of a cycle. With the ATP cycle, you have ATP, which can be hydrolyzed, releasing energy and losing one of its phosphates in this process. A process like cellular respiration can provide the energy needed to add a phosphate to ADP in order to regenerate ATP again, which is important as ATP can be used quickly. This brings us to how ATP is able to work. So how does ATP work? It's not just about ATP being hydrolyzed and releasing energy, it's more than that. Okay, honestly, it's more than our short video can go into, which is why we provide some further reading links, but let's look at some basics. So when ATP is hydrolyzed, meaning here it involves the addition of water, it's not really that the bond between the second and third phosphate itself is a super strong bond. It's actually more that the bond between the second and third phosphate contributes to this ATP being unstable. These phosphates with their negative charges don't like being arranged like this. The change from ATP losing its third phosphate to become the more stable ADP is an exergonic reaction and releases free energy. A popular example for understanding ATP is to use the spring illustration, like a wire spring. Consider how you might compress the spring. ATP would be modeled by that compressed spring, and then you would let it go until it just goes into this relaxed state, which would be represented by ADP. When ATP is hydrolyzed, if the energy was just released, it will likely not be useful for a cell if it's not actually coupled to something that needs it. Thankfully, the energy release can be coupled to endergonic processes that the cell needs to do. This can occur when the phosphate from the ATP is transferred to a molecule that is going to be acted upon. For example, this cell transport protein here is supposed to move some kind of molecule against its concentration gradient. Recall that if it was passive transport, these molecules would be moving from high to low concentration. But in active transport, thanks to ATP, this protein can move them against the gradient. When the phosphate is transferred to this protein, we say the protein has been phosphorylated. Sounds powerful. We can say in our example that this protein is more reactive and less stable in this form, this phosphorylated intermediate state. When it reverts into its original, more stable shape, it can assist in moving them the other direction. So, from marveling at the beating of a single cilia hair, or chromosomes being separated in cell division, or binding the correct amino acid on the tRNA, I could go on. We hope that little ATP symbol will mean something every time you see it. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
There you go. ATP. Got that phosphate, pops off in an exergonic reaction that releases energy that can then be paired with an endergonic reaction that needs energy, like a cell transport protein. And that is the basics of how it functions. That phosphate pops off, passes off energy to some other reaction that's occurring nearby, and fuels every energy requiring reaction that we use in life. All organisms use ATP in this way. And that is all I have for you here. To sort of finish thinking about, remember our definitions of enzymes and coenzymes as the sort of pairing molecules for you today. And then we are activation sites, sort of part of the enzyme that's acting on from an enzyme that's acting on a substrate that is binding to the activation site, and that is or a reactive site, you might call it. And that is our sort of primer on enzymes. That is all I have for you today now. I'll say that again. And I hope to see you in class, and have a good one.